Hello, welcome to the Institute of Biostatistics. This is Kadiru Jayaraman reporting from the Institute of Biostatistics. We had given two lectures earlier on the topic, covering some of the basic concepts and methods. And this time we are going to discuss some of it application in clinical domain. I would strongly suggest you to go through the previous lectures in this regard, so that you shall be able to appreciate the content of this lecture in a better manner. So as a prologue, I would like to cover some of the phases in drug development process. It may not be new for those in clinical domain, but this shall be helpful to people outside the clinical domain. So the drug development process, which would take years together, goes through different stages, namely preclinical, phase 1, phase 2, phase 3, and phase 4. In the preclinical stage, once a potential molecule is identified, it is first tried in animals like rats, rabbits, or monkeys, just to know how safe the drugs are on the animals. When it enters phase 1, trials are conducted on human beings for the first time. They are used more for dose fixing. Emphasis is more on safety of drugs. Involves only small sample size. Because initially we do not know much about the response of these drugs in human beings. So we have to be very careful. In phase 2, what happens is that once we get some idea about the dose level, these are tried on large set of patients, like 50 to 100 patients, and they are done largely for efficacy and safety. So in fact, dosing of drugs is a compromise between efficacy and safety. If the dose level is low, we may lose efficacy, but they may be safe. But if it is overdosed, then we may get efficacy, but at the same time, they may not be safe. So anyway, in phase 2, after phase 2, we may reach a comfortable level of dose level to be tried. So the results of phase 2 are to be confirmed by trying these drugs on a larger number of patients, and like 1,000 to 3,000. So phase 3 trials are more of confirmatory trials and conducted in the regular design, well-designed trials, like randomized clinical trials. After phase 3, the results may be submitted to the drug authorities, approval authorities like FDA, and once the drugs are approved, it comes to the market. So after all, we would have tried the drug only around thousands of patients, but once it is released, it may go to lakhs of patients. And then phase 4 covers the post-marketing stage, wherein signals on adverse events are monitored in that way. It is part of a general pharmacovigilance process, and if some adverse signals are detected to a dissatisfactory level, then it is possible to withdraw the drug that way. Anyway, coming back to phase 1 designs. Now, phase 1 designs are especially amenable to Bayesian research. And we will see soon now. Now, major design types are rule-based and model-based. Under rule-based, we have 3+3 design, and model-based continual reassessment method, CRM, method based on modified toxicity probability intervals, MTPI, and method based on Bayesian logistic regression model, popularly called BLRM. And we will review some of these designs. Coming to 3+3 design, it is the simple-most design to identify a safe dose level. So we start with a cohort of 3 patients with a low level of the dose and observe for toxicity. If it is found to not to be producing any toxic symptoms on the patients, then we may try a slightly higher dose and another set of 3 patients. And again, this process is repeated till at some dose level, we observe dose limiting toxicity. And then at that dose level, we shall repeat the same dose level on another set of 3 patients. And if it is still found to have produced DLT beyond a limit, then we come down to the next lower level, and that would be our recommended dose. So in fact, we can target for different levels of toxicity, but in 3+3, you usually would get satisfied with 1/3 toxicity. That means 1 out of 3 patients show toxicity, or 2 out of 6 patients show toxicity. That too would be considered as a reasonable or admissible level. It is not having any probability concept or any other posterior distribution and so on, like in Bayesian, regular Bayesian approach. But the concept of Bayesian approach is inherent in this process, in the sense at each stage we utilize the past information. Other than that, there is nothing much big about 3+3, but it is very popular because of its simplicity. Next, we come to a regular Bayesian design called BLRM. And for that, first we start with a predefined dosing intervals. Now, we say we identify 3 classes of toxicity intervals, like underdosing, proper dosing, and overdosing. We specify lower and upper limits of toxicity levels, like underdosing 0 to 0.25. 0.25 means once that level of the dose is tried, say 25% of the people show toxicity. A patient may be classified as 1 or 0, depending upon the toxic symptoms are developed on that patient or not. So, but at an aggregate level, we come up, we get a proportion. So similarly, for proper dosing, we may say that the toxicity level in the group is 0.2 to 0.35. Overdosing, anything more than 0.35 to 1, completely all the people showing, the entire set of people showing toxicity could be considered as overdosing. Now, corresponding to these intervals, we may assign some loss value. Overdosing, surely we will encounter dose, and we say we can ascribe a value of 2. Proper dosing, no loss. But underdosing, we may lose on the efficacy side, so there will be a loss. So say, let us say quantify that as 1. So it is not that we are stuck up with these limits. It is possible to modify or choose any appropriate levels as limits. Now, next, what we need is a dose toxicity model. For that, we may choose a logistic function, bivariate logic function, having 2 parameters, alpha and beta. And this function connects basically toxicity level with the dose level. Di star is some reference dose level. And this function, when plotted, would give a sigmoid type of curve. The next task is to specify some prior distribution for the parameters of the dose toxicity model. The parameters are log alpha and log beta, because log that would simplify the nature of distribution. So theta vector could be assumed as following a bivariate normal distribution. So, with mu vector, mu alpha, mu beta, and the variance covariance matrix. So the overall procedure is to first to assign n subjects to the currently tried dose level and make observations on the toxicity. So we apportion these n subjects to certain set of dose levels, and observe the responses, and we fit a dose toxicity model through Bayesian regression, which I have covered in my last lecture. And based on the responses, compute the posterior probability of toxicity at each dose level. Because of the form of the posterior distribution unknown here, we may use iterative MCMC or direct sampling to estimate the parameters. Now, for every steady state iteration, that's what we get through MCMC, a lot of values, and calculate the estimated probability of toxicity using the sample values of parameter theta. Theta is a vector, and that a series of theta values are going to be generated at each dose level, and using those values, we can compute the toxicity level, predict the toxicity levels for each value of theta. So once we get a series of such values, we can partition that as belonging to the toxicity levels we had identified earlier, and then using those proportion of underdosing, then proper dosing and overdosing, we call them PJ, and multiply by the corresponding loss value and sum it over the different toxicity levels, and we get an expected loss, and that is called B of DI. This happens at each level of the dose we have tried. And by comparing these loss values, the dose which minimizes the expected loss is chosen as the next dose. So we try, and I mean this particular selected dose at with another set of patients, and go on repeating this process every time, updating our theta values. Now, when the maximum sample size is reached, the last recommended dose will be taken as the MTD, that is maximum tolerated dose. So now we have seen how Bayesian approach works here. We have a set of patients and their responses to a set of doses, and then we start with a prior and go on improving our belief about the state of the system over time and through classical Bayesian approach. So that is one of the good examples of how Bayesian approach works for phase 1 designs. So the salient point here is that Bayesian approach finds major applications in phase 1 designs in clinical domain, and following our classical case of Bayesian experimental design. So we have not discussed CRM or MTPI, but we have seen how BLRM works here. And is also useful in sample size re-estimation and for controlling error rate in sequential adaptive trials. The adaptive trials is a class of design coming up very strongly in the recent times, and sample size re-estimation is an important topic associated with adaptive trials. So quite often we may feel the need to re-estimate the sample size during the course of an experiment, and based on past observations already collected. Similarly, we may want to stop the trial or certain treatments of the entire set of treatments in the trial. We may stop for efficacy or futility. So such adaptations are possible and are admitted in clinical trials. And we also have the problem of controlling the overall error rate. And in such cases, Bayesian approach has been found very useful. So that's it. Thank you so much for listening. And until we come up with another video, goodbye.